So what do we need to do? Well, first we need to find funding. As Steve said, what I've been doing for the last couple of years now, as you guys send me samples, these samples are building up in my freezer. Because I haven't had funding to spend to generate sequences or to do the DNA extractions. What I've basically been doing is anytime I had a student who was interested in the thing, I'd go to my department head and ask for $100 or something like that so that I could train the student who showed an interest. But as you guys know, the economy has really sort of put a squeeze on us for things like that. And so honestly, in the last year, I just haven't been able to generate a lot of new data, but it looks like we've got now funding in place from the Ozark Chinquapin Foundation to take care of that. What else do we need to do? Well, we need to collect more samples, and you guys are doing this. But I would really like to modify the way that we're collecting a little bit, because I think that if we're going to blow this study up and we're actually going to have a little bit of money to pay to do it the right way, what we really need to do is we need to focus on collecting from populations throughout the entire range. You guys are doing that already and doing a great job. But here's where I think we really need to beef things up a little bit. What I would like to have are 5 samples per population. Now, some populations might not have 5 individuals. They may only have 1 or 3, and that's okay. But if we can sort of average somewhere between 3 and 10 individuals per population, then we'll be able to get a whole lot more information out of the time that you're spending on the ground and the time that I'm spending in the laboratory. As you collect these several samples, several individuals per population, I want you to try to ensure that individuals are widely spaced within populations and we're not picking up, a clone, something like that, right? So if you're working in an area and you know that this plant is completely separate from that one over there, it might be good to focus on spreading these out a little bit. Okay? Once we get all of these collections done, then we need to do the lab work. And Jesse and Taylor are ready to go. Jesse's been chomping at the bit for the last few weeks, and really, I've been the bottleneck. He's wanted to get going on this. We'll do the analyses, and I'll do this along with Jesse and Taylor over the next year. And then what we hope is with this funding coming into place and with a spring right around the corner and a whole new collecting effort, that we'll really have some cool results to summarize for next year's OCF meet. Okay? So I wanted to talk just a little bit about how to collect, right? How can we make sure that what you're doing is in terms of collecting these plants is the best effort that you can give, and how can I make sure that I'm spending the money wisely and I'm spending my time wisely? We need to collect fresh green leaves. Flowers work, mature fruits work, and we need to collect enough of a specimen to make a herbarium specimen. So something like this is pretty good. We've got several good-looking green, healthy leaves, and we've got some flowers here, and that'll make a nice herbarium specimen. And it's also nice green tissue, as you can see, that I can pull. Now, all I really need is about a square centimeter of tissue overall, but, a square centimeter of tissue overall to pull DNA out of. But I need a voucher specimen to go into a herbarium so that if anybody questions the science later, I can say, well, this thing came from this individual specimen. Okay? A specimen that gets to me like this, and this isn't necessarily all your fault, some of this is my fault, but specimens like this I can't get DNA out of, or it's hard to get DNA out of. Older degraded materials that come to me in the mail, some of these I can still get DNA out of, but it's not a great use of our resources, because there's a high likelihood that I'll go through the process, and I'll spend the money and the reagents, and I'll spend the time, the manpower, and that DNA extraction will fail. Okay? And if I try it again, it might work the second time, or sometimes it might work the third time, or something like that. If we try it 2 or 3 times, we might be able to get sort of low quality DNA. But we spent a lot of time in the lab and burned through some money trying to get DNA out of samples that look like this. So, we need to collect fresh green leaves, flowers, mature fruits from a specimen large enough to create a herbarium specimen. I would like to also ask you to send to me, with your specimens, important collection information, like the collector and the date, the state and the county, a locality string, something like shale banks along Highway 6440 just west of Caney Creek, blah, blah, blah. And a lot of our smartphones now will allow us to just go ahead and grab GPS coordinates. That's awesome if you can get that, but a lot of times we can just hop on Google Earth and punch in the locality string and get that. But what I would really like, aside from this basic information, what I would really like to ask you guys to do is to start making some notes on the ecology, right? What are the associate species in the area? And what is the relative abundance of Ozark Chinquapin in the area? Based on all of your sort of incredible experiences with this stuff, what do you think is sort of the natural blight resistance, if any, of the population? Was the population reproductive, right? Were there flowers present? Maybe not necessarily on this individual tree, but in the area. And we already talked a little bit about the morphological characteristics. And ultimately, like I said, what we'll try to do is to tie these ground data to the genetic data in a database to gain a greater understanding of Ozark Chinquapin. All right? And so to tie all of these data together, we need to build a database for the analysis. And here's a database that I've kind of started putting together, my students and I. And so, you know, here's what we call a Q number for Chinquapin. And the Q number is basically just a DNA extraction number. And that's tied to a collector and an identification code. And sometimes you guys have these identification codes on the bags, and other times these codes might not be present. The state, the county, a locality string. It'd be nice to get a little bit more of a locality string from these things so that we could go ahead and generate some GPS coordinates and really do some cool analyses, thinking about genetic distance compared to geographic distance and those kinds of things. And if we do all of that, like I said, I think we're not far away from doing a lot of really cool analyses and learning a lot about this species that all of you guys, all of us, are so enthusiastic about. And so with that, I'll sort of go through this. Here's an overview really quickly, and then I'll leave this slide up. What should we collect? Well, let's try to clip a fresh branch from 3 to 5 to up to 10 separate individuals, separate trees per population. Again, one is okay if you want me to throw it into the mix. I'm cool with that, and especially if it's if there's only one in the area, or if it's a unique tree that you just want to test and throw in. That's good. But after you do that, after you make these clippings, what you should do is place each unique clipping in a Ziploc bag with a damp paper towel. Don't soak it, just a damp paper towel to provide a little bit of moisture. And if you've got flowers and nuts in there, that's great. So put that in a Ziploc bag and then sort of, flatten it like you need flatten it and zip it up to get all the air out so that it's nice and flat, and that Ziploc bag then sort of gives the leaves and stems some structure. If you need to wait a few days before you send it to me, it's great to put it in the fridge. I mean, you think about how long vegetables will last in the fridge, right? You don't want to put them in the freezer. It'd be like putting your lettuce in the freezer and then taking it out and putting it in the mail to me. It's not going to look good by the time it gets to me, right? So put it in the fridge, and that's great. In the fridge, it'll last up to, a couple of weeks even. And I can get it from you, and I can stick it in the fridge for a couple of weeks until I get a few built up to go ahead and extract DNA from. So again, put each unique clipping in a Ziploc bag with a damp paper towel, flatten it, gather the data from the spot, your name, the date you collected it, the locality, state, county, locality string, that sort of thing. Maybe some notes on abundance, ecological data, GPS coordinates, and so on. Again, it's okay to put these in your fridge, but not the freezer. And then put them in the mail to me. And what I was actually thinking about doing was asking Steve to take this and just put it on your website. So it's right. So this stuff is just right there for all of you to.
I'm sorry to interrupt you.
No, no, no.
I'd rather not do that.
Okay.
Because there's so much lost knowledge on this, and I'd say easily, when I first started doing this, 9 out of 10 trees I went to look at weren't in Ozark.
Oh, right,
and,
right.
And the people on the board are the most knowledgeable group, other than
Right.
the people, and the people remember them.
Right.
But I didn't mean to interrupt.
No,
you.
no, no, no. That's what I want. I mean, I want. That's really, I think, the purpose of my talk this afternoon was to really talk to one another and figure out how we can most efficiently get all of the data to me with fresh samples so that the leaves are green and ready to go and in good shape.
What about putting them in a plant press before we send them to you?
That's good too. That's absolutely fine. Yeah, putting them in a plant press and drying them down is just fine. Try not to. I would ask you not to put them in a dryer. Let them dry naturally in the plant press. In the dryer, sometimes if you get the temperature up enough, it can do some things to the DNA.
I just didn't know if the plant press would dry them out too much where you couldn't use it.
No, no, a plant press would be great if you want to do that.
Okay.
Yeah, yeah. Okay, so here's my address. Second day is best, but regular mail is okay too. As long as this stuff is relatively fresh and healthy when you put it in the mail, it should be fine when it gets to me. I would also ask, though, that when you send it out, go ahead and snap an email to me. Steve and I unfortunately learned the hard way. Last fall, I was on sabbatical, which means that I can stay home and write papers and work, but I don't have to go in and teach my class. And so some of us in this job are lucky enough to get one of these things every 7 to 10 years or so. And so I was on sabbatical last fall, and a big box came in from you guys. And my secretary didn't let me know, and I wasn't going in every day. And by the time I did get a call from her and go in and open the box, some of them still looked good, but some had gone south. And so then it's a matter of, do I spend the money and try these things and sort of risk wasting some money, or do I not try it and feel like I've wasted your time in collecting them and sending them to me? So hopefully we can get past that, and that kind of mistake won't happen again if maybe we do some of these things. I gotta say, I felt a little bit better last night at the board meeting. I can't remember who it was. Somebody said something about setting some of these nuts out on a desk and they dried out, right, and sort of lost them, and so it made me feel like at least I wasn't the only one who's had this happen. So with that, I'm happy to take questions.
Oh, no.
Let's see if I can turn the lights up here.
Top, that's fine, that'll work too.
Okay, yeah?
What's the oldest pressed specimen you would take a sample from?
Oh, if it's pressed, a lot of times, if it's pressed in green, it'll work. I have, in my experience working with so my other work is on plums and cherries and apricots, the genus Prunus. And I've extracted DNA from herbarium specimens from the 1930s. And so, that kind of, now, as you go further back in time, some of those things I had to try 2 or 3 times, and so they end up costing more to acquire. But if it really has to do more with what the tissue looks like on the herbarium specimen than the age. If it's still nice and green, sometimes I've tried them even from herbarium specimens from the 90s, but you could tell that the dryer was a little bit too hot and they're a little bit black. That stuff won't work. Yeah. Other questions? Good. Yeah?
What is the approximate cost per sample to go through a DNA test?
So when I think about these things, I typically think about $20 a sample, just sort of roughly. Now, you can save a lot of money by doing them in larger batches and that sort of thing. But I think about doing them one at a time. It's about $20 a sample is what I usually calculate.
What would be that larger batch size that would be applicable to these?
Oh, about 100. Yeah. Yeah. And I don't know that. Yeah, I don't know that I've ever gotten 100 in from you. I think all of the samples I've kept up with in the database, I think over 2 years I'm only at about 100, maybe 110. Is that right, Jesse?
Yeah.
Yeah.
Sounds about right.
Yeah.
So is that a challenge to get 100 cents?
Sounds like you guys got the money, so I got the time.
What's the price break? And how much different?
Oh, it's significantly different. Yeah, half. Yeah, yeah.
Hey guys, we're gonna have to set it all at once.
Good, thank you, everybody.
Okay.